Hello and a very good morning to everyone. Thank you all for joining us today. Hope you all are safe and well in these uncertain times. So I'm Himashri and I'll be your host for today's workshop. I am a senior executive for science communications at the DBT Wellcome Trust India Alliance. So today we bring to you the second lecture of the Continue Biotechnology Education Lecture Series, which is organized by India Alliance in collaboration with the Guru Angad Dev Veterinary and Animal Sciences University, in short GADVASU, and IIT Roorkee. So this lecture is a part of a series which aims to bring to you a diverse range of topics in biotechnology. The first lecture of the series was focused on genetically engineered vaccines. The topic for today's session is next generation sequencing, followed by a session on funding opportunities at India Alliance. We welcome all participants who have joined us today from GADVASU, IIT Roorkee, and many other institutions around the country. So let us first start with a quick introduction to the organization that I represent, the DBT Wellcome Trust India Alliance. So the India Alliance is a charitable trust registered in India that funds research in health and biomedical sciences. So yes, just like the DST, the DBT, CSIR, we do fund research in India. But apart from investing in transformative ideas, we also support research ecosystems to advance discovery and innovation to improve health and well-being. We are funded by the Department of Biotechnology, Government of India, and the Wellcome Trust from UK. Apart from funding research, we fund various programs that promote various national and international collaborations, professional skill building workshops, and also programs on strengthening the research ecosystem. And we promote science education, awareness, and public engagement with science in India. So India Alliance also periodically conducts science communication and outreach workshops as well as public engagement events throughout the year around the country. To keep yourself updated on India Alliance's fellowships, research grants, and many other events and workshops, kindly follow us on the social media handles that you will see on the screen. So more on India Alliance will be shared with you later during the sessions. So before we dive into the main lecture, a few housekeeping matters to keep you aware and navigate the program easily. So this webinar is being recorded. As participants, your videos and microphones will remain switched off during the event to avoid any distractions. You can leave any questions you have for the speakers anytime during the session in the question box. You can also please also ensure that the question only relates to topics being covered in the webinar. In case the webinar abruptly ends due to a technical glitch, please join back using the same registration link that you have used now. Please note that we will be providing e-certificates to those of you who attend the entire session and share a short post-workshop evaluation. We look forward to your feedback after the webinar. And if you do plan on sharing highlights from the workshop through your social media handles, please feel free to tag us. So let me give you a quick overview of today's agenda. So today we have already started with the introduction session. Now there will be a short address by Dr. Yashpal Malik, the Dean from GADVASU. After that, we'll come to the first lecture of the today's program, the next generation sequencing by Dr. Sunil Kumar Mor. which will be followed by a short question and answer session. We'll have a short screen break, following which we will listen and know about the funding opportunities available at India Alliance, which will be given by our grants manager, Dr. Bela Desai, and then we'll have our closing remarks. So without further ado, I would now invite Dr. Malik, Dr. Yaspal Malik, Dean GADVASU, to share a few words. Over to you, Dr. Malik.
Thank you very much, Hima. It is a good day, very good morning to one and all who are attending this very interesting topic, which is being discussed amongst all during this COVID crisis. So I welcome you all to the Wellcome Trust funded and in collaboration that we are organizing this event. On my behalf and behalf of Guru Angad Dev Veterinary and Animal Sciences University, IIT Roorkee, we all three are the partners for organizing this event. And I'm lucky that I'm having the person who has devoted a very long period of research hours working on developing so many innovative ideas in NGS, its applications in detecting the pathogens. That may be virus, that may be bacteria, or anyone that Dr. Sunil Mor is going to tell you during his conversation, during his talk. This topic was chosen because of its significance in the ongoing period of emerging infectious diseases, which some of the times we fail to detect by our conventional classical technologies, the methods which are available, and things have been made easier with the biotechnological approaches or with the state-of-art technologies which are being evolving these days because of the emergence of infectious diseases. Many of them, always we tell, that are having linkages with the animals. They are zoonotic, so there is more interest in the epidemiological surveys, detecting the pathogens which have never been discussed or we don't know about them, how dangerous they could be. And this is one example, that SARS, which is going on, that came from bats. It is not doing anything in bats, but still it has put a very bad situation, a pandemic situation in the whole world, affecting millions of the people. We lost our nearers, our friends in this situation. So we must update our knowledge, and that was the basis of starting this event with the DBT Wellcome Trust, that we will bring some of the topics which are of concern to the public, to the students, to the researchers, so that they can frame some of the research ideas and they can have some knowledge on these topics. So I'm thankful to my own colleague, Dr. Sunil. I think it is a very late evening at his place. Thank you very much, each and everyone, those who have joined and those who are joining very soon this event. And let's proceed with the Dr. Sunil presentation.
Thank you, Dr. Malik, for such a beautiful introduction to the program and how we came about to today's session. So before I call upon Dr. Mor, I'll just give a brief overview, introduction to those of you who do not know him. So Dr. Mor is an assistant professor at the Department of Veterinary Population Medicine and Veterinary Diagnostic Laboratory in the University of Minnesota, St. Paul, USA. He had attained his bachelor's and master's degree in veterinary sciences from the College of Veterinary Medicine at Haryana Agricultural University, and he has further achieved a PhD in veterinary virology and postdoctorate in poultry disease diagnosis from University of Minnesota. So since 2012, he has held several positions at the University of Minnesota and has over 90 peer-reviewed publications, one patent, several book chapters to his credit. Without further delay, I welcome Dr. Mor for his talk.
Okay, good morning. So, thanks Dr. Malik for inviting me for this special lecture and the India Alliance for the invitation. And the next-gen sequencing is a broad topic, and Dr. Hima mentioned that the audience also have the diverse background also from the undergrad to graduate student and the faculty. So I will try my best to cover the broad overview of the next-gen sequencing, and then I will finish up with the examples how we will make decision on NGS application in our research or diagnostic field. So next-gen sequencing, as we know, has impact in almost all the sectors. Even in the space also, we are using the NGS technology for characterizing the microbiome. The sequencing has mainly three categories. The first and the most commonly used earlier time was Sanger sequencing, followed by the second generation sequence, most commonly known as next generation sequencing also, where the Illumina platform is most commonly well known. And then the recent one is third generation sequencing, also known as a long read sequencers. I will explain in the next slide. So both second and third generation sequencing, also commonly known as high throughput sequencing. As we see the revolution in the sequencing field way back in 19s with these long big sequencers, were very time consuming to the nowadays in the recent times with very small pocket size sequencer that we can take to the next to the sample and do the sequencing right away with this samples. And in the coming future, it will be like just a small device we can connect with our mobile also from the export nanopore. It's called SMISIAN. So it's the evolution is it's mainly divided into two categories also. In addition to the second or third generations, they are also known as short read sequencers, the Sanger sequence, and then second generation sequencing are known as short read sequencers because they can go up to 600 base pair sequencing length, while the third generation sequencers are also known as long read sequencers because they can sequence even the entire human plant genome also in just one sequencing run. So in the short read category, the Illumina platforms are well known, but followed by these ion torrent sequencers also. In the long read, the PacBio, which are the big equipments, very expensive, also can only the big lab can afford such sequencers, while the Oxford Nanopore technology have the different types of the sequencers, starting from this small piece, very cheap and easy to afford, to the big ones like Promethean's expensive one also. These are the different companies in the sequencing field. The metagenomics is the broad or the commonly being used term for defining the sequencing of the total genomic DNA present in the any environmental samples. But in the more specific terms, different terms are used for the specific type of the research field. Like if you are working on the viruses, the virome, bacteria, bacteriomes, like that. So next-gen sequencing or this is mainly divided into two components. First and is the wet lab component where we have the sample prep and followed by the library prep and sequencing. And then second important part is the data analysis, so bioinformatics part. So first I will give overview of this, the first part, wet lab part, and what are the different terms related to that part. So if we go with the common term metagenomics, which is like study of the nucleic acid, that metagenomics field or the work can be done in two ways. First, where we know our target. So it's called targeted enrichment method. So in that case, we know our target, and then specific primers are designed like we do for our PCR and Sanger sequencing work, similar here. And then the Illumina, like in case of the short read sequencer, the Illumina adapters are attached, and then do the massive parallel sequencing. So that kind of methods are known as targeted methods, where we know our target and then design our experimental studies accordingly. So one example is in case of bacteria, 16S ribosomal RNA is the highly conserved part, which is in the short read sequencers. Different researchers use either the one variable region or the multiple variable regions. The design primer across the variable regions, and then amplify it, and then do the sequencing. So it's one of the most common method for characterizing the bacterial population in the environmental samples. So like here, amplify the region of interest, and then the index, and then sequencing. While in case of the long read sequencer like Oxford nanopore sequencing, the primers are designed to amplify the complete 16S instead of the partial part, and then do the sequencing. So that's the difference of short and long read sequencers. Second, influenza viruses. Influenza virus have 8 segments, and this genome have the highly conserved part at the UTR regions on the 5' and 3' ends. So one degenerate primer can be designed to amplify all 8 segments. So with this one primer set, we can amplify the whole genome and then do the sequencing. While in case of the long read sequencers, in case of nanopore, they have modified this primer sequence to directly sequence the RNA. So instead of converting into the PCR product where we may add some biasness, the effort is to study the sequence in its native or the natural form. Although that method is still in the initial phase, but that is the beauty of the long read sequencers or this nanopore technology, where we can do the direct RNA sequencing also instead of converting into the cDNA or double-stranded DNA. Another example of again this targeted method, one of this publication on orthohantavirus. So the one method is this non-targeted random primer amplification. Then the second method for enrichment is hybridization based, also known as probe based target enrichment. So in the probe based, instead of doing any PCR amplification, these biotineted probes, they selectively binds to our target of our interest, and then capture those nucleic acid, and rest will be washed out. So only that captured part will be sequenced. The another method is called primer based or the amplicon NGS, where we design several primer sets across the genome of our pathogen of our interest. So like in case here the virus orthohantavirus, but if we are interested in bacteria, all the antimicrobial resistance genes, then design primer across the whole set of the resistance genes. If we are interested in the human part on the exon sequencing, then across the exon sequencing part. Similarly, in the cancer, different cancer genes related the targeted sequencing or amplicon sequencing are very popular. So here, if we see the difference from the previous method, from previous slide, influenza virus or the 16S, only one primer set amplify the target of our interest, while here in the amplicon or the primer based sets, we design multiple primer sets to amplify the whole genome. So this is the primer based method. probe-based or hybridization capture methods are the selective target enrichment methods as compared to the random sequencing method. So if we see the difference of these three methods, see here, if we have the very high virus particles, then we can get good results with random primer sequencing method also. Otherwise, as we go down with the virus particles, like here 10 to the power 2, hardly see any reads for the virus, while in the probe-based method or the amplicon method, still we get very high coverage even with the low viral copy numbers. So that's the beauty of these targeted methods. If we know the sequence of our target, the amplicons or the probe-based capture methods give us more confidence in our sequencing, as well as we can multiplex multiple samples to reduce the sequencing cost also. So I covered the enrichment or the targeted methods. Another method is non-enrichment, non-targeted, or also called as unbiased amplification. So where we do not know our target. So like in case of the outbreak investigation, where we do not have any idea, or in case of SARS-CoV2, when the first sequence came up, there was no prior information what is the virus causing the outbreak. So in that case, we do not have any primer. We just directly add the adapters or the indexes and then do the library prep and sequencing, and then analyze the data to find out what are the pathogens associated with any outbreak. So again, just brief overview of targeted or amplicon methods, where we selectively amplify our product, while in case of the non-targeted method, we just directly do the library prep and sequencing method. So overall follow these basic steps, but with slight modification depending on our experiment or the research setup. In case of the non-biased or non-enrichment methods, although we do not know our target, but if we are interested in Virome study, so we try to reduce the background noise. So like this is the natural form of any sample, where we have the host part, bacteria, and then virus sequences also in very few virus particle compared to the host or the bacteria part. So we selectively do like filtration, nuclease treatment, try to reduce the background noise as much as we can to maximize the reads for the viruses. So that kind of the enrichment method we can apply even if we do not know the, if we are following the non-targeted methods. Sequence data analysis, nowadays and in future, the library prep or the sequencing will be much easier. The more complicated part will be the bioinformatics part or the data analysis, because now, like let's say example of this Oxford nanopore, where we in the couple hours we generate millions of billions of data. So we really need high computational powers and the tools to analyze the data in an effective way, and the better interpretation of that data to extract out useful information from this data. So with that, these are the different steps to follow in any sequence data analysis. We start with the quality check, and then assembly, annotation, and then extract the useful information depending on our experiment. So thankfully, there are the different tools available, some open sources or the commercial tools available to analyze such a huge data. So we have to make the wise decision which software or the tools will be good for our analysis. I will quickly go over some examples of the tools without going much in detail, but just to give you some examples. So like in case of the bacterial whole genome sequencing, these are the different steps to follow. The same like the QC check, assembly, annotations, and then extract out the information, like we are interested in the resistance genes or the virulence genes factors, or the comparison of the core SNPs with the previously available sequences, or the MLST, any type of the information that we are interested, just focus on that. So start with the Unix based command line tools. Nullabar is the one of the most common and well optimized validated data set for the bacterial whole genome sequence analysis, and we are also using it in our lab almost every day. Second is the ASA3P. In that we just upload our sequence and we can get the analysis in different formats. So our job is just to interpret the final output and use that information. MetaRap is another command line tools. Some web-based open sources. Patrick is the one that getting more and more popular for doing these different types of the analysis, genomics, metagenomics, transcriptome, metabolomics, whatever data we have to do the analysis. K-base is also getting more and more useful, and with the options of the different tools, we have just selectively make decision on which type of the tools or the pipeline we need to follow for our data set analysis. And Chan Zuckerberg ID is another open source, which is getting also more and more popular in my experience. This is the most easiest one that I have found to use. Just upload our sequence and then we can get information and do the analysis of the different aspects. The Chan Zuckerberg also collaborated with the Bill and Melinda Gates Foundations. For every year, they have this grand challenge funding where the researchers or the PIs can get experience visiting the Chan Zuckerberg centers, getting training on the bioinformatic analysis, and they also provide follow-up support also throughout the project. Right now, they have the projects in different parts of the world, and I think they advertise every year in December or January. So with this background, where I cover the different types of the targeted, non-targeted, selective amplifications or the enrichment methods, and then different tools for data analysis, then the question is how we should plan our experiment. So in the next-gen sequencing, our common term is garbage in, garbage out. So if we do not have the good quality data, or if we do not plan the experiment very nicely, then we may not get the useful information also. So it will be like wastage of time, money, and our efforts. So next-gen sequencing, as I mentioned, have application in almost every sector or every field. Depending on, we are working on DNA, RNA, protein aspect, we have to decide how to start our experiment, what is our hypothesis, what is our objective, and how to start to plan for the NGS experiment. So this publication nicely summarized the different points of the next-gen sequencing. So I would encourage all of you to read this paper, where they started with like sample type, like which sample type I have to work. Either my sample is the less background noise, like very clean serum type sample, or more background noise, like dirty sample, stool, swab, or in case of SARS-CoV2, the sewerage water is also getting more attention, but it's very challenging sample to process. And then which method do I need to do? DNA extraction, RNA extraction, or total nucleic acid extraction? Should I follow the column extraction or the bead-based magnetic bead-based extractions? And then which method to follow? Should I go with the targeted amplification type or targeted NGS method, or the non-targeted methods? Should I do the positive enrichment, like using the primers based method or the probe based method? Do I need to start my experiment, develop a new method of the primers or probe based, or should I go with the unbiased amplification as a discovery part? So think about all these points in mind, and then what resources I have available for sequencing. Do I have the access to the high throughput sequencers like this very big expensive, or do I have to start with the basic like Oxford Nanopore MinION is just $1,100 to start the experiment. So, and then as I mentioned also, like these different points are very important to think about before starting the NGS experiment, because these are very expensive and time consuming. So again, like we can have different applications. So think in mind whether I'm going for the vaccine purpose or the outbreak purpose or the resistance genes type. So that's more important to start the experiment. So starting with the sample selection, and sample storage is very important, because that is the first point. So if we do not have the right type of samples, good quality samples, we may not get the good quality nucleic acid, and everything will go with that nucleic acid. So this is one study they have proposed, that if we have the same day sample process with the filtration, we may get better reads, viral reads in case of the Virome data, as compared to the archived samples that is filtered and stored at -20 or -80. As compared to the non-filtered samples, we may get very less viral reads. So sample selection, sample storage, and the sample processing is very important to think about. So if we have focus on Virome, we think about reducing the background. If we have to focus on the bacteria or the metagenomics part, we focus on the different process method than the Virome type method. So it's very important to be clear which method or experiment I'm going to do, and plan accordingly. Here is another example of the SARS-CoV2. Once we got the first sequence published, people or the researchers have developed the different methods and targeted methods, because then we can do the sequencing very fast. So the first lab, they have developed this Arctic multiplex PCR, where there was a primer set of around, I think, 300 primer sets, amplify the whole genome of the SARS-CoV2, and so do the PCR, amplify the whole genome, and then library prep and sequencing. So and then followed by this probe-based method were also developed. This is one example of the one company, but I think nowadays almost all the sequencing companies or the different companies have the kits for primer-based or the probe-based. So here is the advantage of, even with this, the MinION, the blacks, the long read or the MI6 short read, even if we go with the very high CT samples, 37 like here, which is almost a negative or very less viral particles, still we are able to get the covering the almost the whole genome with the good coverage of the genome even at very high CT samples. And the sequencing, if we go with the sequencing time and the genome coverage, in very short amount of time, we are getting almost the full coverage depending on the CT of the samples. So these are the another points we need to think about, like the input amount, the turnaround time, like in case of the Oxford Nanopore sequencers, it's the very fast, just 6 to 8 hours as compared to the other methods. And then per sample cost. So like in case the MinION is only $35 versus the Sanger sequencer will cost around $350. So that's the beauty of this targeted method. We can reduce the per sample cost. We do not have to concern about the background noise, and we can do the sequencing at high throughput level. So that is the reason that SARS-CoV2 was one of the virus that is most commonly sequenced across the world because of this targeted method. If we compare this targeted method with non-targeted, so here is the metagenomics non-targeted methods with the different CT samples, and versus the probe-based amplification or the primer-based amplification. So if we see here the difference with the high CT more than 25, we can like have more coverage of the genome. Here is the across the genome with this primer based followed by the probe based, but very inconsistent coverage with the metagenomics or the unbiased methods. So in our project, one of my project on bovine leukemia virus, where we are planning developing the understanding first the transmission of this virus, and then understanding the genomic diversity of this virus, followed by to make some management control methods with that. So BLV have different transmission routes, going from the infected cows to the another cows or the calves. So horizontal or the vertical transmission of this virus. So we started this project and we are doing the sampling of the dairy farms that have the 200 dairy cows or 300 dairy cows, and then almost 50% positivity. So that number of samples we were planning to do the sequencing. So in that case, we decided that we know the proviral DNA sequence of the BLV. So we designed the primer sets to amplify the complete genome of this BLV proviral DNA, and then looking for the faster sequencing method and the per sample cost, we prefer to optimize the method for Oxford nanopore sequencing by following the different extraction methods and optimize the two different labs, optimize the primers and the PCR conditions, and then compare both methods to see if we are getting the consistent result or not, and then do the phylogenetic analysis. So here, see here, we are able to amplify the 8 kb segment in almost all the positive samples. The high CT samples have less amplification, as we see here, with the less intensity, but see the coverage. So we are able to make process 60 samples in just 30 minutes for the library prep and load on the sequencer, and just 2 hours of the sequencing run, we are able to get such a high coverage of the genome. So with this method, we are able to finalize, optimize a cost-effective, rapid whole genome sequencing method, and we are using it now with the applying on all the positive samples, and then do the comparison. Here is the one herd example where this some have the within herd cluster diversity, and then some have the daughter and the dame matching. So we can capture the overall diversity or the similarity of this proviral DNA in the within a herd at different time intervals, and also compare the daughter and dame relationship. So for the vertical transmission or the horizontal transmissions. Another important virus is in the pigs, is causing the respiratory and reproductive disease syndrome, is the PRRS virus. It's a single-stranded RNA virus, but unlike the SARS-CoV2 or this proviral BLV, this virus is changing a lot. So it's fast mutating virus. So this targeted method like probe or primer-based methods may not be applicable. So we rely on non-targeted methods to not to miss any variant or the divergent virus causing the outbreak. So here is the one example of this next-gen sequencing or the whole genome sequencing method, non-targeted method, help us to capture this such a novel recombinant virus that was like showing the identity with the vaccine A virus. So when we did the Sanger sequencing of this region or 5A, it was giving match with the vaccine that was discontinued from the farm, and so that, and also causing the outbreak. So that raised the concern like, okay, what is this virus? Is a vaccine virus causing the outbreak or something new? So we did the whole genome sequencing and find out that this is a novel recombinant virus which has the the backbone from both the current vaccine and the previous vaccine, and some part with the novel part also. So here this bioinformatic analysis is also very important because some beginners, they use the reference-based method, which is more common in the targeted sequencing. But if we apply here, we may miss this recombinant type virus. It may give just hit with the one vaccine and then extract the sequence, pull out the sequence accordingly to give us misinformation on the assembly. Another example is the mouse pox virus. Even though we have the information that by electron microscopy that this is a pox virus, and virus isolation also we did, but the question was whether this mouse pox virus is closely related with the previously published deer pox virus, or it's a novel different type of virus. So again, non-targeted method, even though we have the information of the virus and it's such a large genome of this pox virus, we were able to assemble the whole genome with high coverage. That's one beauty of the NGS method. And then we find the difference at the 2 genes part from the deer pox virus. So we confirm that this is the same species virus with some difference from the deer pox virus. Kind of the same correlation we can do on the human side on the tumor related sequencing, where we just pick up the one particular SNP that may be associated with the tumor. So that's the kind of the beauty of this sequencing with the high depth coverage, even to differentiate at the SNPs level. Another example of we have received this isolate that was causing the cytopathic effect in the cell line, and the lab tried all different methods for the PCR methods for all the commonly known viruses, all came negative. They tried the electron microscopy also, but it was giving some indication that it may be either the aquabirnavirus or the virus. So there was no confirmed conclusion here. So obviously in this type of method is kind of the discovery type. So we can apply the non-targeted or the unbiased amplification methods. So we did that, and we figured out that this is a novel aquabirnavirus. So even the lab have tried this virus PCR based on the previously published sequences or the published PCR assays. They were not able to capture it because this is very divergent. I think it's showing only the 30% amino acid level identity in both segments with the closeless related aquabirnaviruses. So that's the beauty of this NGS on the discovery side. Even if we have such a divergent type of virus, which we cannot capture by any other method, or where these targeted methods may not be applicable, but non-targeted NGS methods can be used. Same another example of this female mallard ducks having this papilloma on the foot and the wing. And in this case, the pathologist was able to collect only the formalin fixed paraffin embedded blocks only. So we didn't get the fresh tissue sample. So if you are familiar with the formalin fixed method, and it's degrade the most of the nucleic acid, not good quality nucleic acid to work for the NGS or any molecular method. But still we work with the whatever sample we have. In this case, formalin fixed paraffin embedded tissue sample, and we did both RNA and DNA NGS method and detected a novel papilloma virus. This manuscript recently published, accepted for the publication. But that's again the beauty of this NGS method, where we can detect the whole genome, assemble the whole genome from highly degraded sample types also. Another example of the, again, unbiased methods, honeybees, where they were the colony collapse, all the bees were dying all of a sudden or losing their wings. If you see this normal versus the infected one, electron microscopy shows small round virus particles, but not able to differentiate what the virus actually is. And with this unbiased NGS method, we have detected 12 types of RNA viruses and one DNA virus. So imagine such a small and but important part of the nature, but it's highly infected with such a high virus load and the diversity of the RNA and DNA viruses. And then followed by we develop the PCR methods to screen the individual sample types and then to know the diversity in the different sample types. So that's first we can do the discovery part, and once we have the background information, then we can develop the PCR method, real-time method, or targeted method to do the batch sequencing in future. One example of on the bacteria side. So for the bacteria, as I mentioned, first we have to think about what is my experiment, what I want to gain from the, what is my objective of the experiment. So some people, they just wanted to capture the microbial diversity. For that case, let's start with if we have the pure bacterial isolate, we can easily go with the any NGS method because it's a pure DNA without any background, and there will be no issue in assembling and sequencing. But if we have the clinical samples, then we have to think about like host will be there, different types of the bacterial DNA part will be there. So which method to apply? So if we just wanted to capture the diversity, then the 16S is the good to classify the bacteria at the genus level. But if we wanted to do metagenomics type work, then we have to do like unbiased methods where there will be a challenge of the host part, also challenge of the different types of the uh, pathogens nucleic acid present in the samples and hence in the sequencing also. So it will be very challenging to do the sequence analysis and interpretation also as compared to this targeted type methods. So I will give one example of the Streptococcus suis. It's a zoonotic pathogen, but it's causing infection in the pigs also. So we are doing sequencing for these pathogens every week, and it has the different strains like pathogenic, opportunistic, or commensal type. So it's very important to differentiate the pathogenic type from the commensal type Strep suis. So by doing the whole genome sequencing, and we just make this final report in a summary form for the pathologist or the veterinarians, where we provide the information of the sequence type, serotype, virulence genes that are selected. But by doing the whole genome sequence, we can also get all the information on the MLST, antimicrobial resistance genes, phylogenetic tree analysis also, one example here also. And that's we can differentiate like this isolate from brain with no meningitis, kind of commensal, versus the brain with the meningitis or septic infection, has the different sequence type or the serotype and the virulence genes pattern as compared to the commensal type. Similarly, for the liver isolate with no lesions, have different pattern as compared to the liver isolate with associated with the infection. So using this information and then the sequence diversity, the companies are also making decision on selecting the isolate for the vaccine, for the autogenous vaccine. And one of our client was able to reduce the infection by 80% in 1 year, just based using this information, and then selectively make the decision on the vaccine strain. So that's the another beauty of this next-gen sequencing and help in selecting the right strain for the vaccine purpose. So as we see by this, the publication presentations on that NGS seems to be very exciting, and it can do almost everything. We just have to do the NGS, but that's not the case. There are several challenges associated with this method. And so the common question that I received in my lab is like, okay, my sample was strong positive, why we're not able to get the full genome. So as I mentioned, if we cannot develop the targeted method for all type of the pathogens, because if the pathogen is very divergent, we may not be able to capture that by targeted methods. So in the non-targeted method, the challenge is that sample has to compete with the other non-related things. So in a PCR, there are specific primers that bind to the target of our interest and then amplify that up to the different levels. While in case of the NGS non-targeted methods, we have to think about like the different components present in the nucleic acid extraction method, and then even after doing some selective enrichment, we may not be able to reduce the whole background noise, or the non-related viruses may also cause problem in case of the non-targeted methods. So here is the one example that was requested for the fecal swab samples requested for the whole genome sequence of a virus. But if you see here, maybe the sample was not collected properly. It has only the plant virus that amplified. So even after putting the million reads, almost 50% of the reads are related to the virus, but it's not the virus of our interest. So it's just the plant virus. So with that, I imagine that maybe when they collect the sample, it was not properly collected, and so that's why we are not able to get the virus of our interest by NGS, even though it was positive by the PCR method. Another method is like another challenge is the non-related viruses. So in the NGS RNA viruses, the single-stranded RNA viruses, they amplify more as compared to the double-stranded RNA viruses. And one example here, like the case was requested for the Rota ABC whole genome sequencing, strong positive for Rota A, followed by B and C. So, but if you see here, the porcine astrovirus was amplified more and having maximum number of reads, followed by the Rota A, and then other type of the picornaviruses are also present. So the Rota B and C have very few viral reads. So it's all consumed by the small picornaviruses or the astroviruses, small single-stranded RNA viruses. So that's another challenge in non-targeted method with non-related viruses also. But on the other hand, it's a plus point if we are looking for the viral diversity or the virome analysis. Then it's good to capture the all different types of the viruses that are present in a sample. In addition to that, we have to think about like whether the virus present in the sample is true or not. So the other term is like adventitious type virus, which is commonly used in the checking the vaccine quality, that there is no unrelated type virus in a autogenous vaccine, or in our case on the diagnostic purpose to make conclusion whether it's a true virus or not. So my lab received some this cell culture isolate from fish. They were suspecting some viruses, but I found this bovine parvo or the pestiviruses in that particular sample, and by doing the interpretation and the further investigation, it came out that it's a serum contamination that was used for culturing the viruses, and that consumed all the reads, and we were not able to detect the actual true fish virus that was growing in the cell line. So the interpretation or conclusion was that this is a contaminant, not a true virus. So that's the interpretation or the data analysis is very important, so that we don't misinterpret or make the misconclusions on the data. So with that, I will conclude that the NGS in our field we use for the diagnostic, but in the research side also I would say have very important application, like without having any prior information like the outbreak cases, we can investigate and find out the information of the pathogen causing the outbreak. High sequence coverage, like in the targeted methods, I gave example of the SARS-CoV2, give us confidence in the accuracy. That's why we can say that, okay, this is the new variant now, and based on just few SNPs change, but we call it a new variants with confidence based on the sequence coverage. High throughput, multiple samples can be processed, multiple whole genome can be assembled, and even the few partial information can be helpful for us to make some important decisions. And the challenge is, as I mentioned, it's very important to collect the good quality samples, appropriate sample type. Data analysis is complex, but it may be more streamlined in the near future. As I mentioned, it's not guaranteed to work with every sample type. Some samples may not be in the good quality to get the good coverage or the sequence data. And in case of the unbiased short read sequencers, they are taking long time, while these nanopore type sequencers are maybe helpful in the future to do the same day testing or in the shorter amount of time. Need to do more validation or the QA quality type work on this NGS procedures, so that we can apply in the research and the diagnostic field with confidence. So with that, I would like to thank all the collaborators and my team members here. With that, I'm happy to take the questions from you.
Thank you, Dr. Mor, for the wonderful presentation.